Okay, good afternoon and welcome to the Proteinopathies ZOOMinar series hosted by University of Michigan. Today, we will host two speakers, Dr. Tom Rothstein and Dr. Miriam Linsenmeier, that will be chaired by Dr. Sami Barmada and by myself, respectively. Please post your questions in the QA box and the chairs or myself will read it for you at the end of the talks. An informal discussion session will conclude the ZOOMinar. Now I leave the stage to Dr. Sami Barmada that will introduce the first speaker. Please, Sami.
Thank you. It's my pleasure to introduce Thomas Rothstein, who's Assistant Dean for Investigative Medicine, Director for the Center of Immunobiology, Chair and Professor of Investigative Medicine at Western Michigan University Stryker School of Medicine. So Tom got his MD and PhD from Duke University, did some clinical training in internal medicine and hem-onc at George Washington University, and also Beth Israel and Harvard Medical School. And then did a postdoc at the National Cancer Institute in Bethesda and also MIT. All before starting his own laboratory at Boston University School of Medicine, after which, being there for several years, moved to Feinstein Institute in New York, where he was investigator and head for Center of Oncology and Cell Biology. But in 2016, Tom made the trip out to Michigan, to Western Michigan University as Assistant Dean for Investigative Medicine. And by 2020, he was appointed professor and founding chair of the Department of Investigative Medicine. So I'm going to focus a little bit on some of the work that Tom has done because it's really fascinating on a subset of antibody producing cells called B1 cells that generate natural antibodies that protect against infectious disease, but also age-related conditions like cardiovascular disease, cancer, and neurodegenerative disease. But today, Tom's going to be talking about something a little bit different, at least I gather, but a highly conserved anti-apoptotic gene called FAIM. This was originally isolated from fast-resistant B cells. And Tom's work, which hopefully he's going to tell about it soon, suggests that FAIM may do more than simply interfere with apoptosis and may have other effects on protein misfolding and the accumulation of misfolded proteins in disease. So I've never met Tom in person, but we've spoken over the phone, and really I'm very happy to introduce you today, and I look forward to your talk. Welcome.
Thank you for that kind introduction, Sami. This is a picture of our 10-year-old medical school set in downtown Kalamazoo, Michigan, about an hour and a half down the road from that better-known medical school at the University of Michigan, where Rams and Sami and others conduct beautiful work in proteostasis. I want to thank Rams and the organizers for giving me the opportunity to present our work to this remarkable group. The phenomenal ZOOMinars I've listened to over the last 2 years have helped me immensely as I try to make a contribution to what's a new field for me. I've spent the bulk of my career as an immunologist studying B lymphocytes, particularly B1 cells, as Sami mentioned. While what I'm doing now may seem like a strange turn, let me explain how this all came about. In the 1990s, there was a lot of focus in the field on activation-induced cell death of T cells. Activated cytotoxic T cells upregulate the expression of a death receptor termed FAS or CD95. Once a viral infection is cleared, these T cells are eliminated by another class of T cells bearing the ligand for FAS. Otherwise, the mass of expanded T cells, the ones that make your lymph nodes enlarge, would increase progressively from 1 year to the next. At this time, we were studying receptor signaling in B cells, and so we wondered whether this activation-induced cell death occurred in B cells as well. And we found that stimulation of B cells through the CD40 receptor induced upregulation of FAS and induced B cells to become very sensitive to FAS-mediated cell death. But activation of B cells through the surface immunoglobulin receptor produced a state of FAS resistance, so that even after FAS was upregulated, if B cells were stimulated through their surface immunoglobulin antigen receptors, they were resistant to FAS killing. this induction of fast resistance required some time to develop and was blocked by cyclohexamide. And so this gave us the idea that there might be new gene expression and new protein synthesis involved. And so we carried out differential display of B cells stimulated by CD40 ligand and anti-IG, that is fast resistant B cells, to see if there was a new gene expressed. And indeed, we identified a previously unrecognized gene encoding a protein of about 179 amino acids and 20 kDa in size, shown here by northern blot. And you can see that it's widely expressed. So we express this gene in a B cell line that constitutively expresses the FAS receptor, BAL17 B cells. And when expressed in BAL17 B cells, you can see that it produced a lower level of sensitivity to FAS killing, as shown by chromium release assay here on the left and by PI staining here on the right. So we termed this new gene FAS apoptosis inhibitory molecule, or FAIM. And just as an aside, the gene that we cloned, FAIM, bears no relationship at all to two other molecules that were confusingly termed FAIM2 and FAIM3. So one of the interesting aspects of FAIM is that we found it has a unique sequence that is unlike any other gene and contains no recognized functional motifs or homologous regions. It's also very highly evolutionarily conserved from worm to human. It also is configured in a unique structure. In early work that we carried out in collaboration with Gerhard Wagner's lab at Harvard, we found that the C-terminal domain in particular consists of a non-interleaved beta sandwich at a relatively unique angle. And later work by another group on a crystallized N-terminal domain showed that it had a similar configuration. This is a picture of the AlphaFold predicted structure, and you can see the two clamshells in the N-terminal and C-terminal regions. As we were examining the genomic organization of this gene, we found that there's an alternatively spliced form of FAIM that's longer by 22 additional N-terminal amino acids. And one of the interesting aspects of this now termed FAIM long is that it's only expressed in the brain. So FAIM short, as shown here, is widely expressed, including the brain, but FAIM long is only expressed in the brain. And I'll come back to that toward the end. So just to summarize what we knew at that point, we had found that FAIM interferes with FAS-mediated apoptosis in B cells. I haven't shown that other work that we carried out over the years showing that FAIM is strongly expressed in germinal centers, is regulated by IRF4, and enhances plasma cell development. But we wondered whether protection against B cell apoptosis is all that FAIM does. And there are a couple of reasons why we wondered about that. One is that FAIM appears very early in evolution in silico. So FAIM appears as early as sponges, actually in conoflagellates, but in point of fact, it appears much earlier than FAS receptor or the FAS ligand. So that made us wonder whether it serves another purpose and only later acquired the ability to interfere with FAS-mediated apoptosis. We made a FAIM knockout mouse, and surprisingly, that FAIM knockout mouse was not embryonic lethal. And FAIM knockout mice live normal lives in our vivarium, despite the high evolutionary conservation that I mentioned, the broad tissue distribution, and the unique structure, all of which would make you think that it was essential for life. In fact, we were so confused by this that we spent $25,000 and made a second knockout mouse, which indeed had the same indolent phenotype. So we considered that protection against fast killing may not be FAIM's primary or sole role. We noted that FAIM is highly expressed in the testis, which is temperature sensitive. And so we questioned whether FAIM protects against heat stress or other types of stress. And to look at this, we started with HeLa cells, and we eliminated FAIM with CRISPR-Cas9, and then subjected these cells to stress, heat stress, as well as oxidative stress produced by treatment with metadione. You can see that these treatments reduced the viability of HeLa cells, but the viability was much more diminished in FAIM knockout HeLa cells as compared to FAIM wild-type HeLa cells. At the same time, we looked at the fate of endogenous proteins in these HeLa cells by filter trap assay. And you can see that aggregated proteins were much more abundant in FAIM knockout HeLa cells that had been stressed by treatment with metadione as compared to wild-type HeLa cells. We also looked at whole animals. We injected mice with metadione and compared wild-type mice with our FAIM knockout mice. We looked at serum LDH as an indication of cell damage and cell death, and you can see that those levels are much higher in metadione-treated FAIM knockout mice as compared to wild-type mice. We also looked at spleens and livers from these mice. We made lysates and centrifuged the lysates with detergent and looked at the detergent insoluble pelleted fraction. And you can see that that's much greater in spleens and livers from FAIM knockout animals as compared to wild-type animals. So at this point, we had found that FAIM interferes with stress-induced cell death in cell lines, in primary cells, which I haven't shown, and in animals. And in its absence, protein aggregates accumulate, that is, aggregates of endogenous proteins. This made us wonder what FAIM might do to aggregation-prone proteins, which are involved in a number of neurodegenerative diseases. And the first one we looked at was SOD1 G93A, which is involved in some familial forms of ALS. So we made fusion proteins of SOD1 and the mutant SOD1 G93A with GFP. When GFP is bound to a protein that aggregates, the fluorescence changes, and so it's possible to detect aggregation on the basis of GFP fluorescence. As you can see, the wild-type SOD1 did not aggregate over a 3-day period, but the G93A SOD1 did aggregate, and the level of aggregation was much greater in HeLa cells that lacked FAIM as compared to wild-type HeLa cells. We showed the same thing by filter trap assay, here staining for GFP, and you can see that it's the aggregated level of aggregated protein by filter trap assay is much greater in FAIM knockout HeLa cells as compared to wild-type HeLa cells. So we then wondered whether FAIM has a direct effect on protein aggregation or whether it's working with other cellular constituents. And so to test this, we carried out a series of test tube experiments. And this is one of our early experiments showing aggregation of SOD1 G93A in the test tube as detected by thioflavin T fluorescence. You can see that SOD1 G93A aggregates over a 48-hour period, and that aggregation is largely opposed by the presence of FAIM. The wild-type SOD1 aggregates to a much lesser extent, and that aggregation is also opposed by FAIM. So we then wondered whether FAIM would have any effect on established aggregates. So we prepared aggregates over a 48-hour period and then incubated them with FAIM for 2.5 hours and looked by thioflavin T assay and by filter trap assay. And as you can see, over a 2.5 hour period with FAIM, there was a decrease in the amount of aggregation for SOD1 G93A, as shown by thioflavin T assay and by filter trap assay. We also looked by differential sedimentation. So established aggregates will sediment after centrifugation and appear in the pellet fraction. After incubation with PBS, that doesn't change. After incubation with BSA, that doesn't change. But after incubation with FAIM, much of the pelleted material now appears in the supernatant fraction. In other words, FAIM has acted on the protein aggregate to change the aggregate to a smaller size, such that it no longer pellets after centrifugation, but appears in the supernatant fraction. But we then wondered whether this was unique to SOD1 G93A or whether it extended to other proteins. And so we looked at alpha-synuclein A53T, which is associated with Parkinson's disease and frontotemporal dementia. And so here is shown a similar experiment to the one that I displayed earlier, showing aggregation of alpha-synuclein A53T as detected by thioflavin T fluorescence over a 36-hour period. And you can see that aggregation is opposed by FAIM long and FAIM short. Here we're testing each of them separately, and they both have comparable activity in blocking de novo aggregation of alpha-synuclein. And then we prepared established aggregates of alpha-synuclein and incubated those aggregates with FAIM short and FAIM long for a 2.5-hour period, and tested by thioflavin T and by filter trap assay. And both FAIM short and FAIM long reduced the level of aggregation as shown by either of these assays. And then we also looked by sedimentation assay, and our aggregates of synuclein pelleted, as shown here, before we started any incubation. After incubation with buffer, they were still found in the pellet. But after incubation with FAIM short or with FAIM long, much of the material is transferred to the supernatant fraction. Here it appears as a tetrameric form, which, as has been discussed in previous talks, the tetrameric form of alpha-synuclein is very stable. And we also looked at amyloid beta, the 1-42 peptide, and again, through a similar series of experiments. Here is shown amyloid aggregation over a much shorter period of time. Amyloid aggregates more rapidly over an 80-minute period. And you see amyloid here denoted AMI aggregating over an 80-minute period, and there's no change when incubated with lactoglobulin. However, incubation with FAIM short or FAIM long largely opposes aggregation of amyloid as detected by thioflavin T fluorescence, and HSP27 does as well. That's been reported previously. And we then asked, as with the previous experiments, whether established aggregates of amyloid beta would be affected by incubation with FAIM. And again, by thioflavin T assay and by filter trap assay, you see that incubation of established aggregates with FAIM short and FAIM long reduces the level of aggregation, here shown by thioflavin T assay and here by filter trap assay. And then similarly, we looked by sedimentation assay. Here, the amyloid aggregates are large and don't enter the gel when resuspended from the pellet. So this is the aggregate at the beginning of the experiment. After incubation with PBS, there's really no change in aggregation. Same after incubation with BSA and with lactoglobulin, as well as HSP27. So HSP27 is capable of affecting de novo aggregation, but seems to have no effect on established aggregates of amyloid beta. However, it's a different story with FAIM short and FAIM long. They each reduce the amount of material that pellets and produce material that is now found in the supernatant fraction. indicating that the FAIM short and FAIM long have somehow disassembled these large aggregates to produce smaller version that can appear now in the supernatant and is resistant to centrifugation and pelleting. So just in pictorial form, at this point, we had shown that FAIM acts on dysfunctional proteins to inhibit de novo aggregate formation and to dissociate established aggregates in vitro. Now, we have no idea how FAIM does what it does because it lacks any activity or functional domains. We're embarking on work to conduct a mutational analysis to understand by alanine scanning what are the important parts of the molecule. This may be important not only in understanding the mechanism, but informing potential therapeutic options that might be available. So to start, we just carried out random mutagenesis with low fidelity polymerase and designed to produce one residue change per protein molecule. And after the first 25 or so such mutations, we found one in which activity was completely lost. So this is S155N, and you can see here shown in black circles is aggregation of amyloid. It's unaffected by incubation with lactoglobulin, and also is not affected by incubation with the S155N mutant. Now, S155 is interesting because, as shown by our earlier work with Gerhard Wagner, this is just at the beginning of a particular loop between beta sheets. And so we hypothesize that this loop may be important in the interaction of FAIM with aggregating protein. We also separated the C-terminal domain from the whole molecule and found that the C-terminal domain has activity equivalent to the whole molecule in terms of interfering with de novo aggregation of amyloid beta. So this is where we are in terms of understanding how FAIM might carry out the activities that it can perform. I want to take a little detour now and talk about a new assay that we developed for protein aggregation. We were a little unhappy with the common assays because many of them provide information about the amount of aggregation, but not the size of aggregates. And some of them, like turbidity, are relatively crude measures, and others require several days to carry out. And so perhaps harking back to our background in immunology, we developed an assay based on monoclonal antibodies. So we used aldehyde sulfate beads of about 4 micron in size that will bind any protein, such as a monoclonal antibody. Monoclonal antibodies will bind a single epitope on a protein. And so if the antibody is bound to the bead after other non-occupied sites are blocked, then a monomer will bind to this antibody by the single epitope which it can display to the antibody, and there'll be no other epitopes for antibody binding. That's different than when the monoclonal antibody binds an aggregated protein. One member of the aggregate will bind to the monoclonal antibody via its epitope, but other members of the aggregate will have epitopes that could bind the same monoclonal antibody. So we come back with the identical same monoclonal antibody in a fluorescent version, and the beads can be read through a flow cytometer, and so we can detect the fluorescent signal given off by the other members of the aggregate to which are bound the same monoclonal antibody in a fluorescent version, and the level of fluorescence will indicate the size of the of the aggregate. In contradistinction, when monomers are bound, there are no additional epitopes available, and so there is no secondary antibody binding and no fluorescence. So let me just show what that looks like. Here is a gel where amyloid is shown aggregating over a 60-minute period, starting out as monomers and ending up after 60 minutes with aggregates that are so large that they don't enter the gel. The microparticle immunocapture assay, or bead assay as we term it, shows a progressively increasing percentage of beads that show any kind of fluorescence, and the level of fluorescence increases as well. So one way that we show this is if we take the 60-minute sample and arbitrarily divide it into 4 quadrants of greater or lesser fluorescence, we can then look at other stages of aggregation. For example, here at 45 minutes, there's not that much difference in the percent of beads that show any fluorescence, but a marked difference in the distribution of the level of fluorescence. At 45 minutes, fluorescence is dominated by the lowest level of fluorescence, and there are no beads that show the highest level of fluorescence. Another way of looking at this is to take the 60-minute sample, centrifuge that, throw out the supernatant, and expose the pelleted material to beads. Take the 30-minute sample, centrifuge that, discard the pelleted material, and look only at the supernatant bound to beads. And the results of fluorescence through the flow cytometer are shown here. We divided, again, the 60-minute pelleted fraction into quadrants and applied those quadrants to the 30-minute sample. There's very little difference in the percentage of beads that are fluorescent, that show any level of fluorescence. And remember that beads that bound monomers are invisible. They don't fluoresce at all. But you can see between the supernatant sample and the pelleted fraction that there's a great difference in the distribution of fluorescence intensity, which means there's a great difference in the size of the aggregates that are bound, where this 30-minute supernatant fraction is dominated by the lowest level of fluorescence and has no fluorescence of the highest or second highest level. So let's focus on the right-hand side of this slide. This shows an experiment in which we took the pelleted fraction and bound it to beads, and then exposed those beads to PBS lactoglobulin or FAIM. We use the quadrant approach to look at the different levels of fluorescence. And so these beads now have aggregate bound to them, and so that aggregate bound to the beads is now exposed to PBS, where there's very little change in the distribution of fluorescence, and lactoglobulin, again, very little change, but there's a great change after exposure to FAIM. There's a reduction in the percentage of beads that are fluorescent at all, but a most marked difference in the distribution of beads, where there's almost no beads containing very highly fluorescent material and only some beads containing the second most fluorescent material. So overall, there's a 50% reduction in beads that have any fluorescence and a 70% reduction in beads that have any of the 2 highest levels of fluorescence. Our reviewers asked whether this assay might work with clinical samples. This is not sufficient for validation, but I'll just point out that we obtained normal CSF and CSF from 2 patients with Parkinson's disease and tested the CSF at 50% and 12.5% dilution. And you can see that the Parkinson's disease CSF samples show more fluorescence according to the bead assay as compared to a normal CSF sample. So there's certainly the possibility that this assay could be useful in terms of looking at the total amount of protein aggregation in CSF from Parkinson's disease patients and in the size of the aggregates located in the CSF of these patients as well. Now, HeLa cells are not neuronal cells, and we initially did not look at neurons, but another investigator in Spain has done so. And this investigator is Joan Comella, and through a number of experiments, he has shown that there is likely a relationship between FAIM and neuronal disease. Here is shown dorsal root ganglion explants, in which he expressed in some of those explants FAIM long, and then withdrew nerve growth factor. And you can see that the explants that express FAIM long persist through nerve growth factor withdrawal much better than those that were transfected with empty vector. He's also looked at the eyes of FAIM knockout mice with increasing age. And he's found, as shown at the bottom, that there's accumulation of protein aggregates in photoreceptors, and that correlates with retinal degeneration with age. And he's also found that there is a reduction in FAIM in hippocampal samples from patients in the brains of patients with Alzheimer's disease, particularly more diminution, a lower level of FAIM at late stages of Alzheimer's disease, suggesting there may be a relationship, a disease relationship there. We've just started our work in this area, looking at neurons differentiated from iPSC cells. So we've taken iPSC cells, eliminated FAIM by CRISPR-Cas9, differentiated them to dopaminergic neurons, and then transfected them using neuromag with an expression vector for synuclein along with fibril seed. And as you can see, in neurons that lack FAIM, there's much more accumulation of synuclein aggregates as compared to neurons that contain the wild type level of FAIM. So this just summarizes in pictorial form our journey from the 90s to the present day in terms of investigating FAIM. And just in narrative form, FAIM is a unique protein with no counterpart in sequence, structure, or function. It defends against stress-induced protein aggregation and cell death in cell lines, in primary cells, and in mice. It prevents protein aggregation of mutant SOD1, mutant synuclein, and amyloid peptide, and it appears to be capable of disassembling established aggregates of the same. We think that FAIM activity toward protein aggregation may constitute a potential new proteostatic mechanism in addition to proteasome and autophagic function. And as the Comella group has shown, FAIM is implicated in neuronal pathogenesis. There are really important questions left to resolve beyond the mechanism of FAIM, whether FAIM's known activities of anti-apoptosis, anti-stress, and anti-aggregation relate to a single mechanism or to separate functions. And then what is the role of FAIM long versus FAIM short in the brain? FAIM short is broadly expressed, including in the brain, but in addition to FAIM short in the brain, there is also FAIM long, which is only expressed in the brain. Why is that? That's an important question to resolve in the future. Overall, we think that FAIM should be renamed. Rather than standing for fast apoptosis inhibitory molecule, perhaps FAIM should be the acronym for fibrillization aggregation inhibitory molecule. And I'd just like to acknowledge the people who've done this work. Tom Schneider isolated FAIM initially. Shimei Zhang identified the long form of FAIM. Hiroki Kaku has done almost all of the work that I showed today and other work which I didn't show today. And Michael Gutnick developed the microparticle immunocapture bead assay that measures FAIM, that measures aggregation amount and size. Gerhard Wagner was our collaborator in the past for understanding the structure of FAIM, and I look forward to collaborating with Sami and Rams in the future. And these are our funding sources, and I'll be happy to answer any questions if I'm able.
Thanks, Tom. That was really that was pretty incredible. I do want to recognize that we have a second speaker, but I'd love to get a couple questions in if we can. So Mercy Best asked, what is the wild type level of FAIM? And in your FAIM dose response bead assay, were the concentrations of FAIM at wild type levels or much higher?
I don't have a number for the wild type levels. In the bead assay for the established aggregates, we had about 22, we had 1 micromolar of amyloid and 8.8 micromolar of FAIM. So there's more FAIM than, but of course this is established aggregates sitting on the beads. The experiment that I didn't talk about where we looked at de novo aggregation, we have 22 micromolar monomeric amyloid and 5.6 micromolar of FAIM. So we flip it when we're dealing with established aggregates.
Okay. Really quick. So I think you hinted at this before. Do FAIM's roles in apoptosis and anti-aggregation rely on similar mechanisms? And would you call FAIM a chaperone?
Yeah, that's a very good question. We're looking at that now. It has no homology to chaperones or HSP27. There's some recent hint that it might work with HSP27, but we did document that when we knocked out FAIM, there's no change in the level of HSP, and so there's no compensatory change in chaperones or heat shock proteins as a result of eliminating FAIM. So I think it's a completely separate mechanism.
Okay. I'm going to try to send you the questions separately so that you can answer them. But I think I want to say thank you again for that talk, and I just want to make sure that we have enough time for the second speaker.
Sure, my pleasure. Thank you.
Absolutely.
Okay, I think that we can go on the discussion at the end of the second talk. I am pleased to introduce the second speaker of today, which is Dr. Miriam Linsenmeier. Miriam has just moved as a postdoc in the lab of Dr. James Shorter. She is giving this talk on behalf of Dr. Paolo Arosio, which unfortunately is not well. Thank you, Miriam, for accepting our invitation. Please, the audience is looking forward to listening to your talk.
So I hope you can see my slides all right. I'm going to go to presentation mode. Yeah, so thank you very much.
It works.
Perfect, amazing. So thank you very much for having me today. Yeah, Paolo is really sorry that he can't give the talk by himself, but he's in the audience and is happy to also answer questions. He just got sick overnight and lost his voice, so I'm here on behalf of him, and I hope I can show you what we're working on in the Arosio group, talking about the interplay between condensation and fibril formation. Let me quickly organize my screen a bit that I can see everything. All right. Sorry for that. Right. So in Paolo's lab, we're interested in understanding liquid-liquid phase separation. Liquid-liquid phase separation has recently been discovered as strategy of cells to form biomolecular condensates and to organize the interior of cells in space and time. But phase separation is actually also important in biochemical engineering. So for instance, in the context of bioseparation, it's important for multi-phase reaction systems and even for formulation of biotherapeutics. And so one important question that we are trying to answer in the Arosio group is, how does condensation affect biochemical reaction and aggregation? And we are doing this by using a variety of different systems. So we have on the one side, these more minimalistic models of biological condensates, so working with real proteins and RNAs. And we are trying to understand the aging of active condensates and also the interplay of condensates and amyloids. And that's something that I would like to focus on today. But we're also trying to learn lessons from these biological systems and design artificial condensates based on simple co-acervation. And so, for example, we are doing this with more bio-inspired systems where we use IDRs that we know are, or intrinsically disordered regions that we know are phase separating, and create by that enzymatically active droplets. And more recently, we're also working with synthetic associative polymers. And so today I would, as anticipated, I would like to talk about the aging of biomolecular condensates, and I would like to work with two different, we would like to explain two different systems. One, where liquid-like droplets age more towards arrested gel or glass-like states, and another, or two other examples where liquid-like droplets transition into amyloids. So in the first system, we were working with an in vitro reconstituted Peabody system. And specifically, we were working with a protein called dead-box ATPase DHH1. DHH1 is an RNA binding protein and is a key component of Peabody's. What is special about it is that when DHH1 is alone in a monomeric solution, it is perfectly soluble. However, when you then add ATP and RNA to the system, you see that there are these really beautiful droplets coming up. They fuse, they're liquid-like, so we have phase separation under these conditions. And the protein is an ATPase, so actually it can hydrolyze ATP, and when only ADP is bound or ADP is in the system, the droplets dissolve. So we were curious how this biochemical or this enzymatic ATPase activity of this protein affects the material properties. And we were doing this, or can do this with different approaches. So first of all, we can look at the material properties using droplet microfluidics. And that's what you can see here on the left side. So we can encapsulate these liquid-like droplets into microfluidic compartments. And what you can see is that these droplets fuse over time to a single condensate that you can then see in the middle of the microfluidic water and oil compartment. And that shows that these droplets are really liquid-like. They have really liquid-like properties. Another approach, another technique that we were using recently, and introducing that basically to the condensate field, is differential dynamic microscopy. And this technique is a light scattering based technique. And essentially what it gives you is an idea about the state of the material state of your system. So you look at you image these droplets over time, and you correlate the fluctuations in the intensity over time from one time point to the other. And what you can see is in the green case, that the system decorrelates fully during the observed time scale, and that corresponds to liquid-like droplets. When you see, on the other hand, something that is depicted here in red, where the system does not fully decorrelate, this represents a more arrested state and could be a gel or a glass transition. And what we can extract is basically relative diffusivities over time. So to understand how biochemical reactions affect the material properties, we were first looking at our wild type DHH1 protein. So this protein, when we add ATP and poly-U as an RNA mimic, forms beautiful droplets, and it is enzymatically active. And what we observe here is that the relative diffusivity remains constant over time. So the droplets are really liquid-like over several days. Only on day 5, we observe this red population of droplets that represents arrested compartments, so that do not decorrelate fully over time. However, when we then used a mutant that is catalytically inactive, so that cannot hydrolyze ATP anymore, we observed that the diffusivity this relative diffusivity is increasing, so we have aging over time. And we also see a big population, a large population of these arrested compartments already much earlier. And so that indicates that really the biochemical activity of these droplets is really important to keep the condensates liquid-like. So just to sum this first part up, the biochemical activity is important for and material turnover of these droplets is important to maintain fluid-like properties of the condensates over time. In the same project, we also looked at LCDs and the structure of RNAs in the turn. I'm not going to show you the data today, but if you're curious, please have a look at the paper. It turns out that also these two modulators are really important to control the material properties. And so we have this kind of this multi-layer regulation that is important to keep the condensates fluid. So in the next part, I would like to look at another type of aging, and this is the aging of liquid-like condensates to amyloids. So these kind of liquid-solid transitions have been described relatively early on in the context of crystallization, where some kind of two-step mechanism nucleates crystals from condensates. And more recently, this has been described in the context of neurodegenerative diseases or in the context of proteins associated with neurodegenerative diseases. So for instance, FAS or agent RNPA1 or alpha-synuclein and tau can undergo liquid-liquid phase separation, and the condensates then go on to some amyloid state. And we are interested in how condensation and fibril formation is coupled in these systems. And I hope that I can give you the two key messages of today's talk, basically. And the first thing that we found is that fibril formation can be promoted at the interface of the condensates. And secondly, that heterotypic interactions can inhibit fibril formation within condensates. So we can actually have both. So in the first part of this part of the talk, I would like to talk about we were working with the RNA binding protein agent RNPA1, and we wanted to understand the interplay between its phase separation and amyloid formation. And as a model system, we are working with the C-terminal low complexity domain here. When we had a closer look at this low complexity domain, we realized that we actually understand pretty well already which types of interactions are actually important for phase separation and also for amyloid formation. It is known from other people's work that phenylalanines and tyrosines in this sequence act as stickers. And so in the sticker and spacer framework, we can actually explain that these residues are really important to promote condensate formation, liquid-liquid phase separation. And then at the same time, this low complexity domain also contains many regions that promote amyloid formation. So those are steric zippers and LARCs. And to understand the interplay of this, we actually generated, so we worked with a wild type low complexity domain that has all of these elements inside. And we also generated a mutant where we deleted 3 peptides that are important for amyloid formation. And we were hoping to generate by this a mutant that would not undergo amyloid formation anymore. So when we first compared the liquid-liquid phase separation of these two variants, we saw that essentially the liquid-liquid phase separation is really similar. You can see in both cases that they form droplets. We encapsulated them again in our microfluidic water and oil compartments. We see them fusing, so they're liquid-like. We also made phase diagrams. We measured concentrations inside and outside the droplets. And it turns out this is all very similar. However, when we look at on longer timescales and we look at the liquid amyloid transition, we see that actually only our wild type low complexity domain is undergoing aggregation. And this is depicted here in the blue curve. This is the thioflavin T signal, which is increasing. And we can also see that by confocal microscopy that after 70 hours, this variant forms these kind of starburst structures and these star-shaped aggregates, while the variant that does not have these amyloid forming regions does not do that. Maybe only a little bit, slightly at the end you can see something, but there is really not much compared to the wild type. So these two variants seem to behave really differently, and this shows that condensate formation and fibril formation are really mediated by different regions of the protein sequence, but they are connected. We also imaged these starburst structures by different techniques. And so, for example, you can see that this happens in a microplate where we can see these beautiful stars everywhere. And also in our microfluidic compartments, we can see them. We also looked at them using AFM. And so on the left side, you can basically see these big blobs and these thin fibrils coming from the droplets. And we also quantified over time the height of the fibrils. And you can see that they're increasing, that this is increasing. So we have fibril growth essentially over time. So then the question was, so what's the purpose of condensation? Now, what does condensation exactly do? And so the first obvious thing that the first obvious effect is up concentration of the protein. So we were wondering, is condensation actually required? And for that, we looked at two different conditions. So first of all, we looked at we took a protein concentration where the initial concentration is below the saturation concentration. So we are actually outside the biphasic region. And under these conditions, the protein does not aggregate within 7 days. However, when we look at the different condition where we are inside the biphasic region, so we are looking at aggregation from condensates, we see aggregation. So either it is a kinetic effect, it is very slow, or we really are below the critical concentration for amyloid formation, we can really not form it. Another thing that we observed, and that is specific to a system that aggregates from condensates, is that the aggregation is basically independent of the initial concentration. So when we start from different concentrations that are where we have condensates, we are inside the biphasic region, we do not see a difference in the kinetics of phase separation. So we might increase the number of condensates and the number of fibrils in the end, but we do not change the kinetics. It turned out in the end that there's another aspect to consider when amyloids form from condensates, and that is that we form actually quite a high number or quite a large amount of surface. And so what we did is we followed the amyloid formation of HNR and PA1 LCD over time. And what we saw is that in the beginning, our thioflavin T dye is just recruited into the droplets, and it's also at a very low intensity. When we then look at around 40 minutes, 60 minutes, 80 minutes, we see that there is this really strong fluorescence occurring at the droplet rim. So there's a THT dye, or the intensity of the THT dye is the highest at the droplet rim. And that told us that fibril formation is really promoted at this interface. So this is unique to a system where we form fibrils from condensates, and we see that the interface seems to have a really crucial role in this. So to look at this a little bit more in detail, we labeled the protein. So we with an ADO dye, and what we can see that is that the protein is up concentrated at the droplet rim already at time point 0, so right at the beginning. So we have already up concentration of protein, but this is not yet thioflavin positive, thioflavin T positive. So this rim occurs only at later time points when we really have the stars. And by contrast, when we did the same with our variant that does not have the amyloid forming regions anymore, we could not see this rim, neither the up concentration at time point 0, nor really the rim at the end point. So this seems to be really something that is specific to this LCD that has the amyloid forming regions. And to make sure that this is not an artifact, we measured the protein concentration kind of across the droplet, and we saw that at the edges, there is a really high concentration of the protein. So confirming our observation with fluorescence microscopy. We also observed that when we increase the NaCl concentration in the mixture, that this rim is basically getting more prominent the more salt we have inside. And what's kind of interesting to see that also at these high salt concentrations, we also have an accelerated aggregation. So also here seems to be a connection. We then asked, okay, if the interface is really so important, when we block the interface, we should actually be able to inhibit or to at least slow down the aggregation of HNR-MPA1 from condensates. And so what we did is we introduced sodium dodecyl sulfate, a really well-known surfactant that should go to surfaces. And when we do that, we see, first of all, that the droplets at time point 0 do not really change. However, over time, only the samples that contain SDS are not aggregating anymore. Basically, we rescue this behavior, and the samples that do not contain SDS are still aggregating. We used a second approach because we actually wanted to see if our surfactant is really going to the surface where it should go, or should be. And so we designed a protein-based surfactant according to a strategy that was recently proposed in this paper. So we designed a construct that has two very soluble domains, this MBP. So you will know that from protein purification, it's a solubility tag, and a GFP. And we attached this low complexity, a variant of the HNR-NP low complexity domain to these two soluble domains. And in this low complexity domain, we deleted all the amyloid forming regions to avoid any promoted amyloid formation. And what we then could see when we added this protein-based surfactant to our BLCD droplets is, first of all, yes, it went to the interface. And when we increased the concentration, we decreased the size. So we basically decreased the size of each droplet. And this is quantified here in this plot right here. And this has consequences on the kinetics of aggregation. And we can basically see that when we have increasing amounts of surfactant, we increase the half-time of aggregation by several hours. So we do not fully inhibit it, but we slow it down. And this confirms that the interface is really important for these liquid amyloid transitions from condensates. And this is really can be kind of described in the framework of these findings from Rohit Pappu's lab that has recently been uploaded on bioRxiv, where they do simulations, and what they find is that there are expanded conformations of the HNR-NP1 protein at the condensate interface. So there are different interfaces, there are different conformations depending if you are inside the droplet, outside the droplet, or at the interface. And these expanded conformation, in combination with certain arrangement, typically more perpendicular to the interface than inside or outside, could promote this amyloid formation and allow for specific interactions of the amyloid promoting regions in the protein. The role of interface in the context of amyloids is it has been described early on. So it is known that the air-water interface, membranes, also nanoparticles can actually have an effect on amyloid formation or on the kinetics of amyloid formation. So for instance, upon protein adsorption on these interfaces, this could increase the local concentration of the protein. It can induce unfolding conformational changes, and in the end decrease the nucleation barrier and promote heterogeneous primary nucleation events. And so just to show an example, this is insulin, and when you add hydrophobic nanoparticles in increasing amounts to monomeric insulin, it aggregates. And depending on how many of these nanoparticles you have, or the more you have of these nanoparticles, the more surfaces you have, and the faster the aggregation. I just want to mention that interfaces can also do the opposite. So basically they can, for example, also induce strong adsorption or restricted mobility, and then it's actually leading to inhibition of heterogeneous primary nucleation events. So it's not necessarily said that surfaces always promote primary nucleation events, but also the opposite can happen. And so I hope that with this part of the talk, I showed you that condensation and aggregation are connected but distinct processes, and that they are mediated by different regions in the protein sequence. Hopefully I could also show you that the interface promotes amyloid formation in HNRNPA1 LCD condensates. And if you want to know more, we just uploaded the preprint, so feel free to have a look. In the last couple of slides, I want to introduce a different system, which is actually even a bit more complicated than what I've just shown you now, where we basically have the same protein forming the condensate and the amyloids. And here we were working with a system where we have a client protein that is aggregation prone, which is recruited into condensates that are formed by a different protein. And so in our case, we were working as the client protein, aggregation prone client protein, A beta 42, because we know really well how the kinetics works. We understand the aggregation kinetics, thanks to the work of Thomas Knowles and Sarah Lindsay, who really dissected all of this. And as scaffold, we were working with IDRs from different helicases, LAF1, DDP1, and DDX4, which formed our droplets. And so by adding amyloid beta, monomeric amyloid beta to condensate, what we first of all saw is that it's beautifully recruited. So it's recruited into these different types of condensates, and it is up concentrated. And initially we were expecting that this local increase of this aggregation prone protein would accelerate the aggregation reaction. But what we observed instead is that it was completely inhibited. So we could not see aggregation anymore. We confirmed that by looking at the residual monomer over time. So this is the residual monomer you have 100% before the aggregation reaction at time 0. And when we then looked after 20 hours of aggregation in the presence and absence of condensates, we saw that when we had condensates in the system, we could basically all the residual monomer was retained and no aggregation really happened. However, when we did not have condensates, amyloid beta aggregated as expected. And this is also confirmed by transmission electron microscopy, where you can see all these aggregates in the absence of condensates and no aggregates in presence of the condensates. This seems to also be concentration dependent. So depending on how much scaffold protein you have, so how much phase separation or protein that can phase separate you have in the mixture, the more you slow down A beta aggregation. So the arrow indicates an increasing amount of sequester or of phase separating protein. So there's a concentration dependence essentially. And at the moment we explain that, or it's very likely that this has to do with some kind of heterotypic buffering effect that has been introduced by the Papu and Taylor labs recently, where we essentially recruit the the amyloid prone protein into condensates, and there we get some sort of competition between A beta interactions and the interaction of A beta with the scaffold protein, and that seems to be enough to inhibit amyloid beta aggregation inside the condensates. And so what we have is that we have some sort of condensates that act as sponges that sequester the aggregation prone peptide and prevent fibril formation, although we have this high local concentration. And so I hope that in total I could show you today that when we have amyloid formation from condensates, it's not only an increase of concentration that promotes this, but that this is actually more complicated. So we first of all have the interface of the condensates that promotes it, but on the other hand, we can also have heterotypic interactions that inhibit fibril formation. And so with this, I would like to thank you all for your attention today. I would like to thank Paolo, my fantastic PhD mentor. I would like to thank everyone in the group who specifically helped with different experiments and discussions, the entire group, all collaborators at ETH and outside ETH, all the funding sources, and I'm happy to take any questions if you have any.
Okay, thank you, Miriam. It was a really inspiring and fascinating talk. My congratulations. Now we can move to the questions that have been posted by attendees in the QA box. First one is by Owen Morris. Very interesting presentation. May I ask a slightly naive experimental question with regards to the in vitro assay involving the dead box proteins that were incubated with the amyloid beta peptide? Were macromolecular crowding agents used in order to mimic the dense environment of the cell and promote the formation of biomolecular condensates? Thanks.
No, I don't think that that they used that there were any crowding agents used. It was really just under the phase separation conditions of the dead box proteins of these intrinsically disordered regions, and then the amyloid beta protein was just recruited to these condensates.
Okay, we can move to the second question from Marcus Drextetter. Thanks for this very nice presentation. Could you comment a bit more on the differences in the aggregation kinetics when using different concentration, but all making droplets?
Yes, I'm just going to go back to the slide. So I guess you're referring to this slide. So this has to do, if you're looking here at this phase diagram on the left side, when you start with the same initial, when you vary the initial concentration and you form droplets, but at the same time you do not change the interaction. So in this case, basically the salt concentration or also the pH, you should basically end up with the same concentration inside and outside the droplets. And yeah, so this basically explains that when you then always have the same concentration inside, that then the kinetics doesn't change. It still can change, like what still can change is the total amount of fibrils that you get in the end, because you have more aggregation prone material in the system, but not the kinetics. I hope this answers the question.
Okay, I cannot see any more questions, but a question from my side. I understand that condensation versus amyloid formations are two closely intertwined mechanisms, but the first one is not necessarily prodromic for the second one. If it is correct, what's the role of kinetics of protein-protein interactions or protein self-assembly in driving one versus another mechanism?
So I think it's in the end, it comes down to what kind of interactions your protein can make. So I think all of these things that we know, like you form beta sheets. So you need something that is competent of forming beta sheets, but beta sheets are not necessary to form to make phase separation in the end. So I think you still need, but if you want to have liquid to amyloid transitions, you still need both elements in the sequence. So you need an element that can promote phase separation and an element that can form amyloids. And I think if you have that, then in theory, it should be possible under the specific conditions potentially, but yeah.
Okay. Okay, cannot read any other question.
Danilo, I think there's a question in the Q&A from Marcus. A follow-up question.
But okay, there is a reply from Marcus, but kinetics are not the same. This is the reply to the previous question. There is some difference in the curves.
Are you referring to this slide
I think
here?
that he is, yes. This
Okay,
question is
yeah, I
relevant.
mean, so there are potentially, like, it's not 100% the same, it's not a perfect overlap, that's probably true. And I think this simply has to do with the heterogeneity of these systems. Like when you introduce phase separation, it's more a heterogeneous system than when you start from a perfect monomeric system. So this might come from there. But in principle, if you look at the lag times, they're very similar.
There is also a comment by Paolo Arosio. I'll read it. There is some noise, but the lag times are rather similar, and aggregation kinetics involved would typically be much more different in the same concentration range. I think that this explains. Okay. If someone would like to raise a hand, can join the panel for further informal discussion.
There's another question in the Q&A from Miriam.
Yeah, I can
Yeah.
see. Yes, I'll read it. Can heterotypic interactions dissolve fibrils?
Yeah, this is a very good question, I guess. I'm not sure if by just adding something, you can really dissolve preformed fibrils that are potentially really stable. But I mean, they're chaperones. They're like that. Some of them involve ATP, some kind of active processes, like you introduce energy into the system, and then it's possible. But I think it will depend on the stability of the fibrils that you have already. If they can be dissolved, and if they're really stable, I think then you need something that is made for doing this, for disaggregating these fibrils.
Okay, we have another question from Marcus. Does deletion of the amyloid promoted regions affect the ability for droplet formation, i.e. not only amyloid formation?
No, so we really tried to investigate that really thoroughly, because we were worried about that of course, if we are deleting parts of the low complexity domain, that this would impact the phase separation. But with any of the assays, we couldn't find any difference. So the droplet had the same size. They had the same behavior with respect to salt, to pH. We also did Raman spectroscopy to measure the concentration of the protein inside and outside the droplets, and also that was pretty much the same. So we couldn't find any real differences.
If I may comment on this, so that would mean that the phase separation or the interactions which drive phase separation are not related to the interactions which drive amyloid formation. Is that correct? Or?
Yeah, so they are distinct, but of course, because the protein can form condensates and amyloids, it's not completely unconnected. But yes, the interactions are different. And it's also interesting when we, for example, when we look at condensation at liquid-liquid phase separation, it's independent of NaCl. But if we increase the salt concentration, look at fibril formation, we see an acceleration. And so what we think is there is one really specific region where a really important aspartic acid is inside. So we think that basically the NaCl can titrate this specific region, or just the charge in the specific region, and therefore promote it. And for as the stickers that are important for condensation are phenylalanines and tyrosines, salt is not really important in this context. And so this kind of experiment showed us that this is really it's independent but connected.
But still, right? I mean, if you say that the stickers are important for phase separation, and you remove them, when you remove the amyloid forming regions, you would assume that you also affect the phase separation.
Yeah. I think we probably just don't remove enough. So I think these
okay.
the stickers are spaced. I think there's always 10, 12 amino acids or something in between. And when we remove hexapeptides, we probably don't remove enough of them that it really has a large impact. So that's what I would assume from the data that we got.
Thank you very much for the very nice presentation. Yeah.
Thank you.
Very data dense.
Okay. we can,
Ron
there are
Staub is
some,
joining the panel.
yeah.
There's one in the chat. Can you ask that question?
Yes, question for Prof. Rothstein from Veena Krishnan. Does FAIM's protein disaggregate activity require energy, e.g. ATP? We cannot hear, sorry. You have to turn on the microphone.
Yeah, sorry. No, it does not require ATP. There's no ATP in the buffer in the experiments that I showed. We speculate this could be a difference with HSP 104, which has been studied by the Shorter lab for many years. That's a yeast protein, but it disappeared in evolution beyond yeast, whereas FAIM has no homologue in yeast, but appeared at the conoflagellate stage. And so this is rank speculation, but one idea is that as animal cells began to move and needed ATP for other things, they dispensed with HSP 104 and acquired FAIM, which can perform a similar function without ATP. But the factual part is that no ATP is required for FAIM's activity.
Okay, we have Martina Pannuzzo in the panel that probably wants to post question directly to one of the speakers. Please, Martina, turn on the microphone. Yeah, go.
Martina is the next week's speaker, so she's in the panel. She may not have a question. So go ahead. Martina, if you have a question, please go ahead. I don't think so.
Martina, can you hear us? No. Apparently, she cannot hear us. Martina?
So no, Danilo, Martina is there in the panel because she's a speaker for next week.
Yes,
Not-
but she
I don't think
posted directly to me a
okay.
question.
Oh, okay.
So I guess that she wants to post some question directly to the speaker, but probably I'm wrong.
Carmelo has a question. I just let him in.
I have a question for Miriam. Droplet formation is just a phase separation, okay? There is a critical concentration for droplet formation, protein concentration, critical protein concentration. Miriam, microphone.
Sorry. Yes, sorry. Yes, so there are, for all of the systems basically that I showed you, there are critical concentrations. And so for HNRNP, for example, this is around 1 micromolar, 1.5, 2 micromolar. Around this, you will not get HNRNPA1 condensates anymore.
Mm. Yes, because 1 micromolar, because EAPP under 1 micromolar non form a fibril.
Yeah, so I think this concentration is really specific to the system. It might differ depending on which protein you have. So like whenever we are working with a different system, we assess this for each protein separately.
Yeah. Sure. Okay, thanks.
I cannot see further questions. Rams, can you see further questions in the Q&A?
I thought there's one in the chat box. Did you read? I'm not sure. The one for Tom.
Okay.
Well, Tom has already replied, I guess.
Yeah, I did.
All right.
Why have not looked at FAIM in PD patients? And I don't think anyone else has this point either. We have IPCC from PD patients and are beginning to look at these things, particularly neurons differentiated from IPCC?
What do you want Tom to address that question? Go ahead, Tom. Maybe you want to.
That was my answer that he read. That's what I wrote. We
I
have not
see.
looked at FAIM in Parkinson's disease patients, but we have a IPSC courtesy of the Michael J. Fox Foundation, and we're beginning to look at levels and the behavior of neurons differentiated from those IPSC.
Looks like there are no other questions. Maybe we can wind up the session. Thank you very much, Tom and Miriam, for great talks. Really appreciate it. Paolo, hope you feel better. So we can have talk maybe in the fall or sometime, Paolo.
No, thank you.
Thank you all very much. Thanks, Danilo. Thank Martina.
Thank you.
Thank you.
Bye.
Ciao.
Bye.